I just talk about that. There are three groups enzymes is very important. This is modification is a reverse and dynamic process. So first, there are two groups of enzymes that can catalyze the addition and the removal of special modifications. And also, histone modifications can function as a platform to recreate the reader proteins, and then it can initiate the downstream pathways. Since the importance of histone modifications during the last 20 years, a lot of evidence to show the association between the abnormal histone modifications and the development of human disease. And based on this, there are some dozens of the small molecules targeting to the histone modification enzymes and reader proteins have been developed as epigenetic drugs for the treatment of so many different cancers and tumors. And recently, there are so many different kinds of new modifications that have been identified. But for those newly identified modifications about their regulation enzymes, regulation mechanisms, and their physiological functions, we almost have no idea about this part. So basically, my research interesting is that we developed many chemical approach to help to decipher the histone modifications from the four parts. First, we need to identify the substrates of the new modifications, and then to explore the biological functions of the new modification. And also, we need to decipher the regulation mechanisms and find the binding proteins to finally to give a whole picture of the histone modifications. First part, to identify the protein substrates of the specific modification, I will use the lysine animation as a typical example to show. So based on a traditional method, based on the antibody-based method, it was reported about 10 years ago, the lysine methylation as a new modification, but at that time, the only 17 proteins were identified as substrates, but it's too far away enough to help us understand the significance of this modification. But at that time, because the modifications is quite difficult to detect, at that time, the specificity of the antibody is not as good, and also the high specificity of the antibody is not available. So deal with the issue, we developed chemical reporters. It is a mimic of the small molecules that is a donor for this modification. So this is a reporter can enter into the cell and then to metabolic labeled onto the target proteins. And finally, take advantage of the chemistry action, we can conjugate the tag. It can be a fluorescent tag or faint tag to the target proteins. So we can visualize the protein, or we can isolate those proteins for identification. So based on this method, we totally identified 40 of the 17 known substrates of lysine methylation. And in addition, we identified another more than 300 new protein substrates of this modification. We did a validation for both the known and the newly identified substrates. It worked very well for all the proteins. And we found that the lysine malonylated proteins are mainly focused in the metabolic process. And this is the method we developed. It's very good for us to fish out those target proteins so we can know who they are, and then we can do further study of their functions. The second part is that based on the structural similarity between the malonylisation and glutathione-CoA, we thought that maybe the lysine glutathione is a new modifications that had not been identified and reported. So to validate our hypothesis, we also developed a chemical reporter, and it worked very well for this modification. We can see a lot of proteins were labeled by this modification, by this chemical reporter, and we found their many proteins were labeled, and there are two proteins here. We are very interested in this part because the molecular weight is around 15 kD. we saw this might be the histones. So we did histone extraction and found that histone can be labeled by the chemical reporter for the lysine glycosylation. Then we did the histone extraction and do the LCMS analysis to find out what kind of specific residues that can be labeled by the glycosylation. Yes, we did find many residues can be labeled by the glycosyl group. And finally, there are total 27 lysine residues can be modified by the glycosylation. And to further study the biological functions of the specific modification, we focus on the glycosylation and the histone H4K lysine 91 residue. We saw this interesting because it's localized between the interface between the dimer and tetramer. We saw that the addition of the negative charge glycosylation will significantly affect their interaction, so makes the nucleosome unstable. But the problem is that based on the traditional method, because of the nature of protein PTMs, it's a very low abundance, and there are the fact that many modifications can occur in different residues at the same time. So it's very difficult for us to extract proteins with homogeneous and set-specific modifications. So from the point of view of the chemical biology, so can we synthesize the modified proteins? And then we can carry out the in vitro study. Yeah, to solve this problem, we perform the expressed protein ligation method. Here, we cut this protein into two parts. One is expressed by the E. coli system, and the other one we can synthesize the peptide with the specific modifications. And then do the ligation, we can finally get the modified proteins with homogeneous and specific modifications. Use these proteins, we then carry out experiment to see whether it can affect the histone optimal assembly process. To do this, we incubate the recombinant and sensitized proteins together, A2A2B, A3A4, and to do the optimal refolding. And then to analyze their folding statement by the set exclusion. Here is the result we have got. It's interesting that the complex contains the modified histone H4 is different from the tetramer and different from octamer. It seems like between them, and it contains all the 4 histone components, but with less amount of the H2A and H2B compared to the wild-type octamer. And also, we want to see whether it can affect the nucleosome assembly process. In addition to the histone proteins, we also add the DNA template to do the nuclear assembly in vitro. And interesting is that we can see that this is wild-type tetrasome only contains the H3 and H4 and DNA, and each wild-type nucleosome contains all the 4 histone proteins and the DNA. But with the addition of the glycosylation, we can see here, we can see the major part of this complex stayed in the tetrasome state, and very little small part can be fully compacted into the nucleosome. So we can see it is quite clear that this modification can unstabilize the nucleosome structure. And to quantitatively study the effect, we use the fatty acid. And here we can see that with the incorporation of this modification, we can see the nucleosome is much easier to deassemble into small pieces into the free histones and DNA. To further study its biological functions in the cells, in living cells, so we need to develop the specific anti-H4K91 glycosylation antibody. First, we test whether this antibody is radio-specific and modification-specific. Good thing is that it's quite good. And then we use its antibody to test their levels. Here we can see that this modification is quite evolutionarily conserved from the yeast to the human cells. And use antibody, we perform the chip analysis to see their genomic localization of this specific histone mark. Here we can see that this mark is specially enriched and the transcription start size, especially for the highly expressed genes. And we also did the chip PCR to do the validation and detect indeed it enriched and the highly expressed genes in the euchromatin, not no expressed genes, not the heterochromatin. And we also identified the regulation enzyme for this modification. The KT2A is a writer enzyme for this modification function together with this complex. This complex can help to generate the glutathione. This is a donor for the glutathione. And we also identified the eraser for this modification, the 37. From the peptide level, protein level, and also the cell level, we can see the 37 can functions as a histone deglutathiolase. It's an eraser for the histone glutathiolation. And since that the histone H4K91, the radio is very important for the DNA damage response. So we test whether this modification is involved in the response to the DNA damage. Here we can say that after treatment with different kinds of DNA damage reagent, we can see the significant reduction of the H4K91 glutathiolation. And it's quite interesting that it is a marker of the gamma H2A phosphorylation is a marker for the early stage of DNA damage. So we saw that maybe the reduction of the H4K91 glutathiolation is very important to induce the chromatin condensation. This is the early step for DNA damage repair. And here, interesting, we can see that if we downregulate the SIR7, we can see the reduction of the H4K91 glutathiolation was inhibited. And we can see the reduction of the signal for the gamma H2AX. Maybe we can say that can prevent the DNA damage repair process. So this is a model we proposed. Maybe the 37 can recognize after the DNA damage. The 37 can recognize the glutathiolation and remove it. And then the chromatin can condense, compact it heavily, and then to initiate the DNA damage repair process. So this is whole picture for the histone lysine glutathiolation. We can see this is a marker for the active genes, and its genetic process is regulated by the KT2A and SIR7. For the third part is that because we think it's very important to identify the regulation enzymes, but it's very difficult to profile all the enzymes because the interactions between the erasers and the specific modifications is usually quite weak and transient. So it's very difficult for us to profile those binding protein if the erasers use traditional affinity pull-down impairment. So to deal with this problem, we developed chemical reporter. We developed chemical probes. So we will focus one typical example is the histone lysine conjugation. And this will report it as a marker for gene transcription. But for their regulation mechanisms, we have no ideas after that. So we focus on the conjugation at histone H3 lysine 4 site. To do this, we developed a chemical approach. It contains 3 essential parts. The first one is the contamination mark and the specific residue. The second one is the photoactive group, and the third one is alkyne function groups. So after the interaction between the binding proteins and the specific modification, we can activate the photocross-linker group to form a covalent bond between the target proteins and the probe. And then finally, we can do the chemistry to conjugate using these alkyne function groups. We can conjugate the target proteins with affinity tag so that we can isolate the proteins for identification. We can compare the method with the select massive cell geometry technique to finally provide the selective and tight binding proteins. Here is the result we got. We can see here these 33 stand out with high affinity and high selectivity. To validate the specific binding between the 33 and the A3K4 constellation mark, we use the recombined 33 protein to do the in vitro photo-cross-linking experiment. Here we can see that only the cross-linked, the put one, only the contaminated probe can selectively capture the 33. And also after the addition of the native A3K4 conjugation peptide, we can see the reduction of the fluorescence signal. And also we can see the with the competition with IC50 around 13 micromolar, and the KD value between 33 and A3K4 conjugated peptide is about 25 micromolar. It was reported that the Xiucong families contains seven proteins, 31 to 37. They were reported as an NAD-dependent deacetylate. AB33 can also function as an eraser enzyme for the lysine conjugation. To test our hypothesis, we performed the LC-mass analysis to see and using the H3K4 conjugation peptides as the substrates. Here the result we have got, we can see that only in the presence of DNA, the 33 can catalyze the removal of the conjugation groups. Here, yes, it is an NAD-dependent histone lysine decontinuase. We also use the histone. We also use the proteins as substrates, and we got the similar result. We can see the reduction for the conjugation level for the whole histone proteins and specific radios and histone A3 and lysine 4. And we also did this experiment in living cells. We can see after the downregulation of 33 expression, we can see the increased signal for the conjugation for the whole cell proteins, and especially for the site, A3K4 lysine site. Finally, we know that the conjugation mark is an active mark for gene transcription, and also 33 was reported to involved in the gene transcription regulation. So we want to down. Here we can see that after downregulation of 3, we can see global enrich and increase of the conjugation mark on the genomic regions of those genes. And safely for several genes, we can see the corresponding upregulation of those genes. So using this method, we can efficiently capture the eraser enzymes, and we finally find the 33 functions as eraser enzyme for histone lysine conjugation to involve in the regulation of gene transcription. The last part is about identification for the histone modification PT-mediated protein-protein interactions, because it's very important. It involves so many different cellular pathways, and it's very important and functions as a drug target for the treatment of many kinds of cancers. But based on the traditional photo-cross-linking assays, we found that it's very easy for us to capture the proteins, but it's very difficult for us to map the binding regions, especially for the proteins with multiple domains. So this is a challenge, and this is a problem we might, because the trypsin, maybe the protein is digested, we can say that the captured target peptide cannot be enriched, even though without the cleavage of the biotin, it can be enriched, but the complex contains the target peptides and the probe peptides. It is too complicated for the data analysis. So deal with the challenge to deal with this problem. We think that we need a further version, a new version of the probe. First, it is photocross linkable, so it can capture the target peptide. Second, it is cleavable so that the probe can be removed away. And third, it is photocross linkable, so that it can be enriched, so make it very clear. So without high background, just we make the data analysis very easier. So based on this requirement, we synthesized a new version of the unnatural amino acid. So this is cleavable, photocross-linkable, and enrichable. So based on this natural amino acid, we synthesized the new version of the propeptide. We think that after UV cross-linking, the protein can be captured, and later, after chemistry and the enrichment and trypsin digestion, we can finally see that only the target peptides can be enriched and cleaved for the data analysis. And this is very small molecule. It's very easy for the data analysis. First, we want to test whether this is a new version of the probe can functions as a new to profile the binding proteins. As we expected, it works much better, even better than before. Here we can see that it can capture so many different the readers and the readers for the H3K4 methylation. And then we continue to see whether it can help us to map the binding regions for the PTM modification. Here, you can see that using as a proof of concept, we use spin-1 and H3K4 methylation interaction as a typical example. This is a reader for H3K4 methylation with the crystal structure available. So we synthesize the peptides and to do the method analysis. Here result I have got the red mark is the labeled residues by the probe. We can see here that all the labeled residues are localized in the binding pocket of the H3K4 methylation peptides. Yeah, it works very well, not only limited in the histone modifications, it also can be applied for the non-histone PTM-mediated protein-protein actions. Here we can see that the labeled residues indeed localized in the binding pocket for the specific interaction with the modified peptide here. In addition, using the recombined single proteins, we also expand our method into the whole cell background. Here we can see that with or without protein overexpression, we can successfully capture the target proteins and map their binding sites. This is a positive controls. This one known as the readers for the H3K4 methylation. We can see the binding pocket. We can see the labeled radius are only localized in the binding pockets for the reader proteins. A good thing is that we identified the new proteins, the CLQBP. It had not been reported to interact histone proteins. So, and interesting is that it contains intrinsic disordered regions. So we think that maybe this can function as histone chaperones. Because histone contains many negative charged lysine and arginines, and this is glutamic acid enriched region can interact with histones and function as a histone chaperone. To test our hypothesis, we first to test whether C1QB can bind with histone H3 and H4. Here is result we can see that after overexpression of C1QB and we did the IP using the anti-flag antibody, we can here see that So histone H3 and H4 can specially enriched by the C1qBP, but not A2B proteins. And also, we use this protein to do the in vitro tetrasomal formation assay. We can see here that with increased amount of the C1qBP, we can see the increased formation of the tetrasome. This is a wild type tetrasome. We can see here this is this appear as a position of with similar with the wild tetrasome. So we can see that we can confirm that C1qBP can function as a histone chaperone to facilitate the tetrasomal assembly process. And in living cell, we want to see whether it had any potential functions. So we did the C1qBP knockdown and use the luciferase as a reporter gene. We found that with the depletion of C1qBP, we can see the downregulation of the gene transcription. Further impairment may be needed to further study the specific and detailed mechanism how C1qBP can function as histone chaperones. This is further a plan for our research. And finally, so this is a method we have developed. It is a trifunctional amino acid can help us to map the binding regions for the protein PTM mediate protein-protein interactions. So finally, we'll summary. We are interested in to decipher the protein modification from the 4 parts. For the protein substrate identification, biological functions explanation, and study the regulation mechanisms, and to study the PTM-mediated protein-protein actions. Finally, I will thank all my collaborators and all my lab mates and students, and finally, I thank you all for listening.